Hello, this is Gina Piscatelli with the second lecture on the lymphatic system for Anatomy and Physiology 2 at Madison Area Technical College. In this lecture, we'll examine the functions of specific lymphatic organs, tissues, and cells that live in those organs and tissues. To begin, let's remind ourselves of the organs associated with the lymphatic system. There are primary lymphoid organs and secondary lymphoid organs. The primary lymphoid organs include the red bone marrow, where all leukopoiesis takes place. So this is where all lymphocytes and other white blood cells are born. And the other primary lymphoid organ is the thymus gland, and that's where a specific type of lymphocyte matures. So it's necessary for T lymphocytes to function. I think that's why it's considered a primary lymphoid organ. You'll see the importance of T lymphocytes when we discuss the immune system, as well as a little bit at the end of this lecture. So the primary lymphoid organs are red bone marrow and the thymus gland. The secondary lymphoid organs, the lymph nodes, spleen, those two organs house leukocytes, lymphocytes included, and are sites where infection can be controlled. The lymphoid tissues are aggregates of leukocytes and connective tissue surrounding them in the tonsils, the appendix, the large intestine, the respiratory tract, and mucosa in general. So the mucosa, what mucosa means is the inner lining of a hollow organ. And there's a lot of lymphoid tissues associated with openings to the external environment. So you can imagine this would involve the digestive system, the urinary system, as well as the respiratory system. So the functions of primary lymphoid organs include leukopoiesis, which is where the red bone marrow has stem cells that differentiate into white blood cells. And most of the white blood cells mature in the bone marrow. In fact, B lymphocytes do, and this drawing shows you that B lymphocytes leave the bone marrow, enter the bloodstream, and can go populate a lymph node. Okay, now there are some cells, lymphocytes, some types of lymphocytes called T lymphocytes, and for them to become mature and able to function, they leave the bone marrow, travel in the blood, but they need to go to the thymus first, where they learn how to recognize foreign antigens, and I'll talk about that in a second, but this is where they become mature. From the thymus, once they've passed their test and they're mature, they can go populate a lymph node. Okay. So those are functions of the primary lymphoid organs. Let's talk a little bit about what I mean by maturation. A leukocyte becomes completely mature once it's completely differentiated or fully differentiated, meaning it contains all the enzymes, granules, or organelles that it needs to perform whatever its function is. Now there's a lot of different types of leukocytes, monocytes, eosinophils, basophils, etc. In the lymphatic system, we focus mainly on lymphocytes and macrophages, which are derived from monocytes. So I'm going to focus on lymphocytes because that's what we talked about in the previous slide with the bone marrow. A lymphocyte, a specific type of leukocyte, becomes mature when it becomes fully differentiated, containing enzymes, granules, and organelles, like mentioned before. But it has two further requirements to be fully mature. And that is that it needs to be self-tolerant, meaning it has to be able to recognize our own cells and learn to ignore them so that it doesn't, fight our own body systems. It also needs to become immunocompetent. And what that means is that it needs to learn, each cell, each T lymphocyte or B lymphocyte needs to learn to detect only one foreign antigen, not multiple foreign antigens. So we have a set of lymphocytes that recognize smallpox and another set of cells that recognize a rhinovirus. And so depending on the infectious agent, you'll have different lymphocytes that respond, not the same lymphocyte. And we'll talk about that much more in the immune system, but I wanted to explain why the thymus is listed here. It's because this is where T lymphocytes learn to become self-tolerant and immunocompetent and fully mature. Okay, now we're going to look at secondary organs. And this picture shows a dissection of a human cadaver beneath the diaphragm. And the diaphragm is right here. So we're looking at the abdominal cavity. And the stomach has been deflected a little bit so that you can see the pancreas, which lies underneath it, and the two kidneys, so the intestine's been removed pretty much. The two kidneys here. And this is the aorta. And here's the inferior vena cava. Notice how the inferior vena cava is wider in diameter than the aorta is. That's exactly what we talked about concerning arteries and veins, is that veins are capacitance vessels because their diameter is wider. Anyhow, back to what we're supposed to be talking about, the spleen. So the spleen is tucked up here near the diaphragm in the left side, and it has a really rich blood supply. And we're going to see why that's so important and how it aids its functions. So the functions of the spleen are twofold. One, there's some functions that relate to immunity. And then there are other functions related more to storage or being a reservoir. Recall that the spleen is the site where red blood cell lysis occurs, and that macrophages that are present in the spleen, macrophages are white blood cells, can remove the hemoglobin from a red blood cell, separate the heme out and the iron out so that the iron can be sent to the liver to be stored. So it's involved with, I guess, the storage of iron in the sense that the heme is going to be sent to the liver to become bilirubin, and the iron is going to be sent to the liver to just store the iron. And although storage doesn't happen in the spleen, you need the macrophages in the spleen to separate those things. The immune-related functions relate to the fact that there's a lot of lymphocytes present in the spleen, and they can proliferate there, meaning divide, increase in number, and enter the bloodstream from the spleen. Therefore, they not only survey the blood coming into the spleen, but they can go take up residence in other places as well and survey the blood. So we say that the spleen is a site for immune surveillance of the blood. There are macrophages there that can clear foreign matter and damaged or old erythrocytes, leukocytes, and platelets from the blood. And this picture that I'm showing you in the lower right here is the histology of the spleen, what the spleen looks like under the microscope. And there's regions that are dark, and there are regions that look lighter. Funny enough, the names are kind of opposite, and that has to do with the staining. So the region that's dark is called white pulp. The region that's lighter is called red pulp, and that reflects the type of cells that you would find in each location. So in white pulp, that's where you'd find the macrophages and the lymphocytes. You'd find the white blood cells. And in the red pulp, you'd find red blood cells. Now, I've talked a lot about how the spleen is a site where red blood cells are broken down and the parts are harvested for storage or whatever. But what's really cool about the spleen, I think, is that this red pulp is a site where healthy red blood cells can be stored. So we have a pool of red blood cells not circulating in the blood located within the spleen. I think that's really cool because I'm going to show you another slide here. So just to be clear, the histology reflects this dual function that the spleen has. We have the white pulp where immune surveillance takes place, and then we have the red pulp. Well, I don't know, spot to choose. I guess I'll choose over here, but that's where erythrocytes are destroyed, but then also stored. Slide should say where erythrocytes are stored. And so let me show you a healthy, freshly harvested spleen that has been cut so that you can see its interior, and you can see that there's a capsule surrounding the outside. That capsule we call the external capsule surrounding the spleen. It can contract and force red blood cells into the circulatory system. So this is a really important function, particularly with sudden blood loss, right? So some sort of a trauma and you lose a lot of red blood cells, the spleen can contract and a bunch of red blood cells can be dumped into the circulatory system to carry oxygen. It's really cool. Okay, let's talk about other lymphoid tissues. They are prevalent throughout the entire body. Some of these tissues are diffuse with a loose arrangement of white blood cells, and these are the ones that are associated normally with the mucosa of the digestive tract and the respiratory tract. But some tissues are nodular, and so there's more of a dense aggregate of the white blood cells, and they appear like spherical bodies, or like organs in and of themselves. These nodular lymphoid tissues include the tonsils, the lining of the appendix, and the lining of the small intestine, where the tissue is referred to as the lymphoid tissue is referred to as a Peyer's patch. So let's first look at this loose arrangement of lymphoid tissue, mucosa-associated lymphoid tissue, otherwise known as MALT, to abbreviate it. So this picture is an example of diffuse lymphoid tissue, mucosa-associated lymphoid tissue in the large intestine. And where the black arrow is pointing is an aggregate of loosely aggregated white blood cells. Okay, so anywhere you have an opening to the exterior environment, you'll see at least MALT. You'll see mucosa-associated lymphoid tissue with white blood cells aggregated in locations like the respiratory tract, the reproductive tract, urinary opening, the digestive tract. Although in the digestive tract, we have the tonsils, and then we also have the intestine, which has mucosa. Not so much the mucosa-associated lymphoid tissue. The mouth doesn't really have much other than the tonsils. So now we're talking about more dense or nodular lymphoid tissue associated with the digestive tract. these tonsils contain lymphocytes and crypts that trap bacteria or foreign particulate matter. And so these tonsils can gather and remove many of the pathogens by the actions of the lymphocytes that enter the mouth, the pharynx, with food or inhaled air. So there's two tonsils that you'll find in lab later that I'll mention here. One is the pharyngeal tonsil, because this is the pharynx here. The pharyngeal tonsil, and when they're really enlarged, you might hear clinicians call them adenoids. So if somebody has their adenoids removed, they're having their pharyngeal tonsils removed. And then there's also a tonsil kind of at the back of the roof of the mouth called the palatine tonsil, because you know where the palatine bone is. So these used to be removed when they became inflamed, but that doesn't happen as much anymore. Not that I'm a clinician, just a human. But anyway, let's look at some other nodular tissue in the small intestine. There's lymphoid tissue that's considered nodule or nodular. I don't know, I'd call it MALT because it's associated with the lining of the small intestine. But whatever its classification, in the small intestine, these aggregates of white blood cells are called Peyer's patches. And they are arranged kind of in a follicular sort of pattern, similar to what we see in the tonsils, but they're really noteworthy. It's in the submucosa. Okay, the next part of the lecture deals with the functions of the cells that reside in all these tissues and that also function within the immune system. So lymphoid cells, what kinds of lymphoid cells do we have? They're derived from leukocyte stem cells, and they function within lymph nodes of the lymphatic system or as part of the immune system in other organs throughout the body. So they're found in virtually all organs and tissues, but concentrated in the organs and tissues we've already looked at. Two of these cells are shown in this photomicrograph, a lymphocyte and a macrophage, well, two lymphocytes and a macrophage. Notice that both of these cells are located on a network of reticular fibers. We just call them reticular fibers. This tells me that they're probably this picture was probably either taken from a tonsil or a lymph node, maybe the thymus, but I doubt it because I just know the structure. Regardless, this slide just tells you that there are cells that are going to function in the immune system, but they're located within the lymphatic tissue. And we're doing the lymphatic system. So we'll cover the cells. So the different types of cells that we will talk about are macrophages, dendritic cells, and three different types of lymphocytes. So first, the macrophages. These are cells that were derived originally from monocytes formed in the bone marrow. When monocytes leave the bloodstream and enter either lymphatic organs like lymph nodes or systemic body tissues to fight infection, they turn into macrophages. They differentiate. They mature in another way. So like monocytes, macrophages, you could think of them as daughter cells of monocytes, perform phagocytosis. And that process, phagocytosis, is also known as cell eating. Maybe you've heard of that, cell eating. Now the dendritic cells are located in the submucosa of the skin. And they also perform a kind of phagocytosis, sort of, but they use receptors to bind to foreign cells or foreign agents. rather than engulfing. And I'll show you that in a second. Then lastly, there's 3 different types of lymphocytes. T lymphocytes, named as such because they mature in the thymus. B lymphocytes because they mature in the bone marrow. And finally, natural killer cells. So let's look at the T lymphocytes first. Some of the T lymphocytes are what we call cytotoxic T cells. What this means is that they can attack and destroy foreign cells directly. Other T lymphocytes we call helper T cells, because what they do is activate other immune cells. I know that sounds a little bit, lower class, so to speak, but actually helper T cells are paramount in forming memory cells that provide us with protection when we get vaccines. You have to have helper T cells activated. So we'll talk more about those later. The second type of lymphocyte is the B lymphocyte. These are the cells that produce antibodies that recognize a specific pathogen or foreign antigen and tag it for destruction, or tag it so that macrophages will recognize it more easily and phagocytize it. Finally are the lymphocytes called natural killer cells. These cells attach to and destroy altered host cells. And these altered host cells might be cancer cells or virally infected cells, unlike, a completely foreign cell like a bacteria. Let's look at how macrophages perform phagocytosis. We can see the process in this picture. First, the pathogen adheres to the macrophage. So that's what the pathogen is green in this picture. The macrophage is purple. And then the macrophage extends out its cell membrane and cytoplasm to engulf that pathogen, eventually breaking off into a vesicle that we call a phagosome. And this phagosome then is located on the interior of the cell within the cytoplasm. Now, in the third step, the phagosome fuses with a lysosome. So here's the lysosome that already existed inside the cytoplasm. And when these two things fuse together, the enzymes in the lysosome break down that pathogen. So the pathogen's going to get broken down. Eventually what happens is that the phagosome with the residual parts of the pathogen, this phagosome will fuse with the cell membrane, and through exocytosis, that residual material will be expelled from the cell. So that's the process of phagocytosis. Dendritic cells work a little bit differently, I guess. I don't know if it's that important that you understand completely, but I do want you to realize that a pathogen that's recognized by a dendritic cell will bind to a receptor on the dendritic cell. And on the opposite side of the membrane where the receptor exists are specific molecules. Here it's called a coated pit. The molecules are called clathrons, which you don't have to know. But from here, a similar process happens, right? So the pathogen binds to a receptor, and you get the formation of a vesicle on the inside of the cell containing that pathogen. It just happens to be bound to a receptor. But it's still going to fuse with the lysosome. And so here we have it fused with the lysosome. Now, though, it's called an endosome instead of a phagosome, whatever. But things don't really even out, meaning it really almost stays as two different vesicles, in that this part of the vesicle will degrade the pathogen and eventually undergo exocytosis. But the part of the vesicle that still has the receptor will then fuse with the cell membrane so that receptors are recycled, so that the receptor can be used again. But you can think of dendritic cells as kind of cell eating, but the process is called receptor-mediated endocytosis rather than just phagocytosis. Okay, so that takes care of macrophages and dendritic cells. Now we're going to look at lymphocytes. And let's look at the first two types of lymphocytes first. The cytotoxic T cell and the helper T cell. Both types contain a receptor called a T cell receptor, which is this funny kind of rectangle on the outside of the cell. And that T cell receptor is specific for only one type of antigen. But these cells also have other membrane proteins. And you'll notice that labeled on the cytotoxic T cell is a membrane protein called the CD8 protein. It's usually close to the T cell receptor. And the helper T cell has a CD4 protein, also located close to the T cell receptor. The importance of these different CD proteins will be addressed when we cover the immune response later in the semester. What is important at this stage is that the cytotoxic or CD8 T cell binds to a foreign, cancerous, or infected cell and directly kills it. That's why it's called cytotoxic. And the helper T cell, or the CD4 T cell, when it recognizes and binds to a foreign antigen with that T cell receptor, it'll release chemicals. And those chemicals will recruit other white blood cells to help fight an infection. And I've already mentioned that it's this helper T cell which is really responsible for increasing the number of lymphocytes that are specific for a particular antigen, and therefore forms the basis for which we can fight off serious infections and achieve memory in our immunity once we've been exposed to a pathogen. So I can't stress that helper T cell enough. So the only thing that's new really on this slide is the concept that there are these other proteins, that there are proteins on the cell surface of T lymphocytes. And one that's very important is the T cell receptor, because that's what's going to bind to whatever is foreign. The other one that's important, the CD proteins, will become apparent later in the semester when we do the immune system and how it works. Let's just show you a picture of how a cytotoxic T cell can directly kill a cell, though. That's kind of cool. So the T cell receptor binds to a foreign antigen on the target cell. We'll say this yellow cell is a foreign cell, and it's got some kind of foreign surface molecule that we'll call a foreign antigen. And the T cell has a receptor that recognizes that, so they bind together. In response, the cytotoxic T cell is going to release these molecules called perforins that make a pore in the target cell. And then, if it's not bad enough that you've got holes poked in the outside of your existence, your cell membrane, if that's not bad enough, it also deposits enzymes called granzymes into the cell. And those granzymes will start degrading molecules on the inside of the cell and trigger apoptosis, which is known as programmed cell death. And so then what you have is a dead target cell. That's what you have. The helper T cell also binds to a foreign molecule with that T cell receptor. So here's the T cell receptor down here. The foreign antigen I labeled, the red one, I labeled it here in red. Now that foreign antigen might be on a cell, as is shown here. It also might be freely suspended. But once that binding event occurs, then the helper T cell releases all sorts of chemicals. These chemicals have various names: cytokines, interleukins, interferons. But in summary, all the molecules that are released will activate or recruit other white blood cells. For example, recruiting neutrophils to the site of an infection, a bacterial infection. Activating natural killer cells. We haven't talked yet about them, but they work just like cytotoxic T cells. Activating macrophages and alerting them to come to the site to perform phagocytosis. Activating eosinophils to destroy any parasitic toxins. And activating basophils to release histamine and promote inflammation or an increase in blood flow to the area. So that's one thing that's really important about helper T cells. There are other things that we'll talk about with the immune system. So here I just wanted to tell you that the last kind of lymphocyte, called a natural killer cell, functions just like the cytotoxic T cell in that it uses perforins and granzymes to kill a target cell. But this picture is a little bit misleading, so I want to go through it with you. On the upper left is a very small natural killer cell releasing perforins when it has recognized an enemy cell. Then in the upper right, you see that the natural killer cell now has released granzymes into the enemy cell, which is going to trigger apoptosis, which is shown in the lower left. So here's the enemy cell, and it looks all poofy now. I think that's supposed to signify that it's starting to degrade. It's the last picture that is a little misleading, the lower right. In the lower right, the enemy cell is tiny now, and the natural killer cell is gone. It has left the scene, and a macrophage has come along to engulf what remains of the enemy cell. I wanted to point that out because if you look at it at a glance, you won't notice that how the natural killer cell left the scene, I guess. Okay, now we're going to look at B lymphocytes. And these lymphocytes mature in the bone marrow. They produce antibodies. These antibodies recognize foreign molecules that are freely suspended in body fluids, or the foreign molecules might be displayed on a cell. Now, what do these antibodies do once they bind? They do bind to these foreign antigens. What can they do? They can neutralize an antigen, which is really helpful if it's a toxic molecule, or they can tag it for destruction so that a macrophage can more easily recognize that this is a foreign substance or cell that needs to be engulfed. So that's what antibodies do. And that is really the end of our lymphatic system. We'll talk more about immunity after spring break, I think. Thank you very much for listening.